Mighty, let's continue our session. It's so relevant that ordinates make good indicators of freshwater ecosystem. Consequently, we compiled a list of ordinator species recorded to date in the Isimangaliso Wetland Park. Isimangaliso Wetland Park is identified as an important area for ordinator diversity and endemism, a trend also reflected by the DBI values. Let's stay tuned and know more about it. Good afternoon, everyone, and welcome to yet another session of National Symposium on Conservation of Ordinates, jointly organized by the Department of Zoology of St. Joseph College for Women, Alappuzha, and Department of Zoology of Vellalar College for Women, Erode, under Office of Academic Collaboration in association with Society for Ordinate Studies, Kerala State Biodiversity Board, and DBT Star College Scheme, Government of India. I cordially invite Ms. Ramya James, Assistant Professor and Head, Department of Zoology, St. Joseph College for Women, Alappuzha, Kerala, for the welcome address.
Thank you, Ermina. Respected dignitaries and participants, I am so glad to welcome you all to the third session of National Symposium on Conservation of Ordinates. With great pleasure, I welcome principals of both college, respected Dr. Rita Koto, St. Joseph's College for Women, Alappuzha, and respected principal Dr. S.K. Jayanti, Vellala College for Women, Erode, to this session. And today's session is handled by Mr. Vivek Chandran A., Conservation Biologist, Society for Ordinate Studies. About Mr. Vivek Chandran, he did his post-graduation in Ecology and Environment Sciences from Pondicherry University. And he has around 6 publications to his credit, and also is part of 3 books, publication of 3 books. And he is having work experience in as assistant grade 3 in Food Corporation of India, and as junior technical assistant in Central Warehousing Corporation. And he has gained experience in management of insect pest from both these institutions. And presently, he is a PhD scholar in the Department of Geology and Environmental Science, Christ College, Iringalkuda, Thrissur, Kerala. And he is also having expertise in GIS and remote sensing. Warm welcome to you, sir, to this session.
Thank you.
Also, I'm extending warm welcome to all teachers, research scholars, and students from different parts of India. Also, gratitude and welcome to the head of the Department of Zoology, Dr. Indumati D., Vellala College for Women, Erode, and other members of the organizing committee, and the coordinators, Dr. Kalaivani, Department of Zoology, Vellala College for Women, Erode, and Sujit V. Gopalan, Expert Consultant, Society for Ordinate Studies. Thank you so much.
Thank you, ma'am. Now, let's lend our ears for the valuable talk given by our today's resource person, Mr. A. Vivek Chandran, Conservation Biologist, Society for Ordinate Studies, based on the topic Diversity of Ordinates, will focus on wetland. Over to you, sir.
Thank you, Arpana. I'll be presenting now. Hope my screen is visible to you all.
Yes, sir.
And I genuinely hope nobody is in the mood for a siesta after having lunch. That's what people usually do. That's what I usually do, because in the mornings I will be in the field, in the wetlands, how it is. Have you been to the wetlands? Any wetland recently? It's very hot in Kerala. I'm pretty sure it's the same in Tamil Nadu also. So once you go into the field and you stay there from from 8 o'clock to 11 o'clock, and then you come back, then you have lunch. I know what it feels like. So I, I usually take a nap at this time. So forgive me if I talk a little sleepishly. But I'll try to be talking, about ordinates, so it will be very interesting for me. So I hope it's the same for you. This is what I love doing. First of all, I'd like to thank the organizers of this symposium for inviting me, because this is what I love doing. Go to the field, take photographs, observe ordinates, and come back and talk to people like you, with the hope that at least a handful of you will get interested. So let me start my presentation. So we'll just run through what a wetland is. I'll just show you some big pictures. That's all. I just want to keep you motivated and engaged. because this is the time of the day when people find it hard to focus. But these are the things that you already know. This is just something that I took from Wikipedia. You know what a wetland is, right? Yeah. So how is it different from any other freshwater aquatic ecosystem? So all wetlands have this something called seasonality. Okay, because the water level changes according to the seasons. Yeah. So the bird diversity changes because there are migrants coming and going. The same with the fish diversity. So we know that it's the same with insect diversity also, because we do have migratory insects, including dragonflies, coming to our wetlands. So why do we study wetlands? What is its significance? So yeah, I think this picture summarizes it in a very nice manner. So because almost all the wetlands have been brought under the management of human beings. So we depend on them. We are so dependent on them, but still a lot of wildlife has managed to coexist with us in the wetlands. So this picture that is taken from coal wetlands, I think it summarizes what a wetland is, or the condition situation of our wetland. Our wetlands are rich in biodiversity. That is the first and foremost thing. And I don't need to emphasize what is the importance of diversity. Right from our food to medicine, we depend on biodiversity for everything. Okay, and in the case of wetlands, there is something called Ramsar sites. I hope all of you are aware about Ramsar sites. Ramsar Convention was a convention that was signed by many countries internationally at a site called Ramsar, which is in Iran. So at present, we do not, our Ramsar sites in India do not enjoy any legal protection, but it is the duty of the government to conserve these wetlands. So we have many such wetlands in South India, out of which this coal wetlands is one. And I'm pretty sure there are wetlands like Kalliveli Lake. I've been there in Tamil Nadu, Kalliveli Lake, Usteri Lake. Such wetlands are Ramsar sites. These are some of the conditions that the sites have to satisfy to be classified as a wetlands. There is an overemphasis on birds here, because birds are the very charming organisms. They migrate, they're migratory, and they are so dependent, especially the water birds are so dependent on our wetlands. It is the same case with the dragonflies, but not many people have bothered to study dragonflies and highlight their importance. So that is why most of these criteria are dependent on birds. So any site that has over 20,000 water birds, any wetland that has over 20,000 water birds is classified as a Ramsar site. It qualifies as a Ramsar site. Otherwise, if there is more than 1% global population of any water bird species in a wetland, it qualifies as a Ramsar site. Okay, this is just for your information. This is another picture taken from the coal wetlands. And we humans, we depend directly on wetlands for food, either for paddy. We are all South Indians and we depend on rice, as our staple diet. And we also eat fish. So all these come from wetlands. And then they recharge groundwater. The wetlands, they act as huge tumblers of water. And the water soaks in, and basically your wells have water because there are wetlands around. Otherwise, your wells won't have water, and you'll be running around for getting drinking water. So that's one thing. And then we humans have managed wetlands in such a way that we are able to manage floods. For instance, you can see a barrage that has been constructed here. This is again in coal wetlands. It controls the quantity of water that goes into the sea. So this flood is under our control now, thanks to these wetlands. And yeah, the wetlands have aesthetic value also. Where else can you just take your bicycle and go for cycling just like that? Enjoy the nice breeze, see some birds, spend some good quality time. You can do the same in a forest area, but you need special permissions for that. But if you have a wetland nearby, you don't need anything. You just need to go there and spend some time, and you'll have a nice time. You'll feel more relaxed. So that's also very important. It does have a tourism potential also, because I personally feel the sunset looks much prettier in a wetland than in a beach. Okay, I don't know how many of you agree, but that's what I feel. I've been to the beach. I've been to many wetlands. So this is what I feel. But do we value these wetlands? That the wetlands have all these qualities, right? They satisfy our need for food. They satisfy our aesthetic needs, everything. But do we value them? Are we valuing them properly? Because in this picture, you can see, you have everything here. You have these birds, migratory birds here. Yeah, you have rice growing here. And you have this vegetation. But in the background, you can see constructions. These are basically encroachments. They're being filled up illegally. So this is happening. This is actually happening. These are the photographs that I've taken when I go to the field. So this is not from some other country that you're seeing. This is happening to our wetlands, just right around us. So so now to come to ordinator diversity. Why do we study ordinates? That's a different thing that I think with 2 or 3 sessions that were taken the last 2 days, I think you are very familiar with ordinates. But in Kerala, I'm not sure about Tamil Nadu. From Kerala till date, we have recorded 175 species of ordinates. I can say that Tamil Nadu is data deficient when it comes to ordinates because there are not many people observing ordinates in Tamil Nadu. So this is the right opportunity for you guys from Tamil Nadu. I sincerely urge you people to observe ordinates around you because we truly don't know what ordinates could be found around you. In Kerala, at least in one district, at least we have 3 or 4 people observing ordinates, but that is not the case with Tamil Nadu. We have very few people interested in ordinates. Okay, so it's the right opportunity for most of you to start observing ordinates. In the Western Ghats, we have 193 species. In India, we have over 500 species of ordinates. So when you compare the diversity of ordinates with other insects, for instance, butterflies, you can say that it is very less. We have in Kerala, we have over 320 species of butterflies identified till date, but we have only 175 species. Why is that so? One thing is that this is a very ancient lineage. Many of the species have been lost. It doesn't show much of diversity as such. As in, the ordinates are by nature, they show some moderate diversity only. They're not as diverse as other groups of insects like Coleoptera or beetles or butterflies. But this is also an advantage to us because we don't need to study many of the ordinates. It's very easy to identify ordinates because we have only a few species of them. We'll just look at some of the species that are found in the wetlands, and why we should study and conserve them. Okay, yeah. This most of you would have seen in the last 2 days. Yeah, I'm pretty sure many of you would be able to easily identify a dragonfly from a damselfly. I would like to keep this open to questions and interactions. If there is any question you can ask in between or at the end, I would really love to answer some of your questions. I don't want you guys to just watch this as a movie and forget everything after this is done, because that is not the purpose of it. I truly want at least a handful of people to get interested. Yeah. So dragonfly and damselfly, what are the basic differences? Dragonflies are robustly built, and most of them keep their wings open, when they're resting. But the damselflies, they're very delicate, and most of them keep their wings closed and close to their abdomen when they're resting. And look at their faces. The dragonflies have huge eyes that they touch each other. Well, damselflies have smaller eyes and they're wider apart, at the 2 sides of their heads. That is the basic difference. We'll look at the differences later on. And then if you are to study ordinates, you need to know some basics of taxonomy. Okay, but even if you don't know any of this, even if you do not know what a head is, what a tail is, even if you don't know any of this, you can easily identify at least some 80% species that are seen around you. That is the ease and that is the charm of dragonfly watching, because they're so easy to identify. Many of the species, especially the species that are seen in the wetlands, they're very easy to identify to the species level, even to the species level. But what about the remaining species? For that, you may need to study some of these parts, body parts that you need to know about their morphology. That is the basic requirement. So like any insect, a dragonfly has a head, a thorax, and an abdomen. It can be broadly divided into 3 parts: head, thorax, and abdomen. And the abdomen, it looks like a train, you see. As a train has many compartments, it has many segments. The abdomen has many segments. And the second segment, in the case of male dragonfly or damselfly, a male ordinate, it is very important because that is where in the second abdominal segment, you have what is called the secondary genitalia or the mating organ. The mating organ is located here, but the sperm producing organs, okay, the primary genitalia are situated in the 10th segment. And these are not connected internally. So the dragonfly, the male dragonfly or damselfly will have to bend its abdomen and transfer the sperm to the secondary sexual organ before mating. That is something that you would have seen already in yesterday's and day before yesterday's presentations. And at the end of their abdomen, at the tip, at this 10th abdominal segment, you have what is known as anal appendages. The anal appendages are well developed only in the males because it is used by males only, So it is functional only in the males because it is used for grabbing the female by its head or prothorax. So that is what is shown here. Okay, so that is very important because in the case of some confusing species, anal appendages will have to be examined to be identified for the species to be identified to the species level. Okay. So because for each species, the structure and coloration of the anal appendages would be different. So this is something that you will be familiar with. Yeah. And why are they known as ordinata? I hope Balachandran sir would have talked about it, because the term ordinata comes from odonto, because they have very sharp teeth. They have biting and chewing mouth parts, like the cockroach, and they are very fierce predators. Okay, so this is their head. This can be compared to our face. This is the nose. The nose portion is known as frons. Below that you have mustache that can be termed as clypeus. Then you have your upper lip, that is labrum, and you have your lower lip, that is labium. In the case of odonnates, the labium is 3-lobe. It is made up of 3 parts. So if you cut a dragonfly's head and watch it under a microscope, it will be like a monster. Like a predator, alien or something, because its lips, the lower lip will have 3 lobes. It's made up of 3 lobes. Okay, and most of the head is made up of eyes, you see, because dragonflies and damselflies, that is how they perceive their environment. Their eyes are very, very important. Their antennae are not well developed, so they don't really have a very good sense of smell. How they perceive their environment is by vision. That's why the eyes are very important, and each of these eyes are compound eyes made up of over 30,000 lenses. Okay. And in the case of ordinates, the wing venation is also very, very important for classifying them. Their classification, at least till the genus and family level, are dependent on the wing venation. Each of these veins that you see here have names. Okay, but don't get worried, because as I told you, over 90% of the ordinates do not require these wing venation or facial markings to be to identify them to the species level. We just by looking at birds, you can say it's a crow, or it is a myna, or it's a parakeet. No. Just like that, you can identify most of the ordinates that you see around you, especially in the wetlands. But how do you differentiate a male and a female? This is the very first step in ordinate taxonomy. What is the first difference? Okay, these are the 2 pictures of this is a male, this is a female. They belong to the same species. What is the difference that you see here? Okay, the male is very, very colorful, right? Very colorful, very flaunty. Like in lions have manes, no? Only the male has mane. And in the case of peafowl, only the male has the feathers, beautiful feathers that you call train. Okay, just like that, only the males have these very good, very bright coloration. There is a reason for that, because males are the ones who compete among themselves for gaining access to the female. So they need to be very showy. They need to tell the other males that I'm very colorful, I'm very showy, I'm very, I'm very powerful. That has to be shown. So that is why the males are very colorful. And they also need to attract the females. So this coloration really helps them to do that. But in the case of females, the condition is opposite, because they have to carry eggs, right? So they cannot fly. That weighs them down. Each of these female carry around 300 eggs. So it's not easy to fly with these eggs. They act as a baggage for them. So they cannot just fly away and fastly, just like the males do. So the females have to be very cryptic. So that is why they do not have bright colors, and they should not, because they should not attract the attention of predators. They should not basically attract unwanted attention. So their behavior is also very cryptic, because they will be seen near water bodies only during the mating time. Otherwise, they won't be seen. What you see near water bodies are mostly males, because they will be there guarding their territory and attracting males. Okay. So if you like, this is the secondary genitalia that has been highlighted. Okay, this is the second abdominal segment. You can see a small bulge here that is seen only in the male. In the female, that plane is plain. That place is plain. See, they do not have the secondary genitalia here. Okay, their genitalia are situated at the end of the abdomen. But in the case of males, the primary genitalia are situated at the 10th abdominal segment, and the secondary genitalia is situated in the second abdominal segment. And in the case of males, the anal appendages are well developed. You see this? But in the case of females, they are just stub-like. They are not at all well developed, because they do not have any function. Only the males grab the females. So the anal appendages act like fingers for grabbing females. Okay, in the case of dragonfly males, you can see 3 anal appendages. 2 of them are superior and 1 is inferior. But in the case of damselfly males, you can see 4 anal appendages. 2 of them are superior and 2 are inferior. That is the basic difference. And in the case of damselfly females, you can see an organ called stylus here. Stylus is basically a sense organ. It works for the sense of touch. It is for testing if a substratum is good for egg laying. Okay, just behind this sense organ called stylus, you can see the ovipositor, which is like a spear. It is used to make holes in the aquatic vegetation to lay eggs. So that is seen only in the damselflies because they are the only endophytic oviposition. They are the only ones who do endophytic oviposition. There is one exception in dragonflies that I will come to it later. But most of the dragonflies lay eggs directly on water surface. So they don't need this ovipositor. So this ovipositor is mostly seen in damselfly females only. Okay, now let's jump straight to it. We'll just see some damselflies first. And in the case of damselflies especially, you have to look for the male for a species level identification, because damselflies, as I told you, the females are very cryptic. They are very difficult to spot. And even if you spot them, they all look alike. It's not easy for a beginner to tell apart the species by looking at the damselfly females. But in the case of males, it's relatively easy. So let's take a look at this family called Lestidae. They are in English, they are called spreadwings because even though they are damselflies, they have their wings wide apart. When I told you about the differences between dragonflies and damselflies, I told you that most of the damselflies, when they're resting, they fold their wings and keep them close to their abdomen. But in the case of this family, the family Lestidae, they're called spreadwings because they keep their wings spread despite being damselflies. Okay, and they're usually seen in vegetated ponds in wetlands. So these are some of the common species seen. This is the commonest of this species. This is called Emerald Spreadwing, Lestes elatus. In the case of damselflies, we mostly refer to, even for dragonflies also, we mostly refer to scientific names because like birds, we do have common names for dragonflies and damselflies, but they are not very much in use and they can get very confusing. Because and from the same alternate, if it is seen in India and in some other country like Sri Lanka, they'll have a different common name in Sri Lanka from what we have here. So that is why we depend mostly on scientific names. Okay, just keep that in mind. This is Lestes elatus. It is called Emerald Spreadwing. In the case of this damselfly, by just looking at its thorax, you can say that this is a species because you can see this hockey stick-like marks, green-colored hockey stick-like marks, that is not present in any other species. So this tells you that just by a single glance, you can say that this is a Lestes elatus. And in the case of this species, Lestes primorsus, it is also seen in vegetated ponds in wetlands. It is mostly bluish in color with black markings, and its legs are brownish in color. So that is enough to tell that species apart. And here I have shown a magnified drawing of this anal appendage of Lestes elatus. So these are the larger ones are superior anal appendages, and what you see below them, the smaller stub-like things are the inferior anal appendages. So you do not really need to see this image, of the anal appendages to tell these species apart, but it does come in handy in certain species. Okay. Now we come to this family called Caloptergidae. These are not very regularly seen in wetlands because they do require some shade. So they're more common in forested areas, but they do occur in some of the wetlands, especially if there is some shade. Okay, so how do you know a species belong to this family? Just look at their legs. Caloptergidae. I'm sure in Tamil also, Kalu means our legs. No, so the legs are very long here. See. For these damselflies, the legs are very long. So that's how I remember that they belong to Caloptergidae. They are broad wings. Look at their common name, broad wings. You see, most of the damselflies have very narrow wings, but in this case, in this family, they have very broad wings. Okay, that is also there, and most of them look like their bodies have been embedded with metallic green plates. So that's a very important and very easy way to identify these species. Okay, so we'll take a look at our two common species here. They all look alike in their coloration and their structure, general structure, but look at their wings. So one is clear-winged forest glory, and as the name says, its wings are clear. But in the case of Vestalis apicalis, that is black-tipped forest glory, the tip of the wings have a black shade to it. So these are very easily distinguishable in the field. Then we have species known as Chlorocyphidae. So this is also very easy to identify in the field because you see most of the damselflies, when they keep their wings closed, their abdomen is longer than the wings. But in this family, the wings are longer. See, none of the families have this peculiar character. This is the only family of damselflies, Chlorosiphidae. Stream jewels, they're called stream jewels because they have these, very bright coloration in their wings, metallic coloration that looks like LED bulbs. That they can reflect sunlight. That is why they have this very peculiar markings on their wings. Okay. So this is not very usually seen in wetlands, but if your wetland is nearer to a forested area, you may find them in the streams. This is known as stream ruby. And another species is this southern heliodor, this Libelago indica. It can be easily distinguished from the stream ruby because of its coloration on the wings. It doesn't have any metallic coloration or bright coloration in the wings. It has only a reddish shade on its wings. Okay. Now we come to platic Nimididae. These are, in English, they are known as bamboo tails. Some of them are called bamboo tails because their abdomen, which is wrongly termed tails, they're not actually tails, they're abdomen. So they look like bamboo sticks. That is why they're known as bamboo tails. Okay, so these species are very difficult to identify in the field, and for that you need to look at their anal appendages. You see this? In the case of Copera vittata, the superior anal appendages are about half the length of the inferior appendages. But in the case of Copera marginippus, the superior appendages are very, very small, almost as if it is not there at all. So that is how you identify these two species. Now we come to the most common species, that is a Ceriagrionidae. They belong to the family Ceriagrionidae. They can be found everywhere, right? From ponds, streams, forested areas, to even in towns you can see them. So they are the most adaptable damselfly species. And this is one of the commonest species, Ceriagrion occidentale. It can be identified because look at its common name. It's called green striped slender dartlet. So in between these two black marks on the thorax, you can see a light green colored stripe on the thorax. So that is a very easy key to identify the species, Ceriagrion occidentale. But as I told you, in the case of females, these markings may not be very clear. The female appears very dull in color and mostly plain in color. So it is very difficult to identify damselflies by looking at the females. You need to look for the males. And then this is a very interesting species because it is called Kerala dartlet. Okay, Agonoptimus keralensis. It was discovered in 1984 from Kerala. And after that, it has been recorded from Karnataka, Goa. So basically, we assume that this species occurs only in wetlands adjacent to the Western Ghats. It has been recorded nowhere else. Okay, so this can be easily identified because of this second abdominal marking, this marking on the second abdominal segment. That is this O-shaped marking. Okay, similar to that, we have the pygmy dartlet known as Agonoptimus pygmea, but you can tell it apart very easily because in the case of Agonoptimus pygmea, only the last 2 or 3 segments are orange in color. But in the case of Agonoptimus keralensis, almost the entire abdomen is orange in color. Okay, I'm talking about only the males. You see here, the females are also shown here, but the females, it's very difficult to tell them apart. They all look alike. So I'm focusing here on males only. So these are some of the very common species found. Ceriagrion species. Okay, I'm sure many of you would have seen this species, but you would have brushed it aside. I don't think many people would be interested in dragonflies. But I hope it changes with today's class. Okay, so you have the Coromandel marsh dart, which has its face and its thorax greenish in color, and its abdomen is bright yellow in color. But in the case of Ceriagrion chromothorax, this is a recently described species which was discovered only in 2019. Okay, so it has its thorax also yellowish in color, and there is of course a difference in anal appendages. Okay, so we have another Ceriagrion species, which is very easy to identify because it is called orange-tailed marsh dart. It has 2 orange spots, right? One where its abdomen starts and one where it ends. And in between, the abdomen is black in color. So you don't have any other species like this in India. So it is very easily distinguishable. Ceriagrion cerenorubellum. Then you have 2 species of Ischnura. Okay, these are smaller damselflies and they look alike. I'm talking about only the males, but even the males look alike except for their abdomen. In the case of golden dartlet, which is also called the Western golden dartlet, the entire abdomen looks yellow, bright yellow, except for the last few segments which are sky blue in color. But in the case of Senegal golden dartlet, even though the abdomen is yellow, it has a black stripe over it, and the last segments are sky blue in color. Then we come to a species known as Pseudagrion species. These are wetland dependent species. but their taxonomy is very complicated, and it is very difficult to identify them without looking at their anal appendages. These three are different species, but if you look at them like this, they appear the same, right? There is not much difference. But if you look at their anal appendages, their structure is very different, and they belong to different species. Now we come to dragonflies. Okay, in the case of dragonflies, first of all, we come to this group called darners. They're not scientifically termed Aeschynids. Okay, this is an exceptional family because when I told you about how dragonflies lay eggs, most of them lay eggs on the surface of water bodies, right? Except for this family. In the case of this family, the females have this ovipositor, the spine-like ovipositor that I was talking about. All the damselflies have it, and only this family in dragonflies has it. That is called ovipositor, and the family is known as darners. Okay, they are very large dragonflies, sometimes called helicopters, because they make that whirring sound of helicopters, and they're very large in size. And if your wetland is located somewhere near the forest, you can easily see this known as blue darner. Its face is blue in color, and it has its thorax has blue and black markings, and they're very huge in size. They can be easily distinguished. But look at these two species. These are wetland-dependent species. One is Anax guttatus, and the other is Anax indicus. At one look, they may appear the same, right? But they do belong to two separate species. Just look at their markings on the abdominal segments. In the case of Anax guttatus, the markings are pale yellow in color, and they're separated. But in the case of Anax indicus, the markings are yellowish in color. They're bright yellow in color, and they seem to be joined together, the markings. So this is how you can distinguish these species. And then you have something called as Kaduva Thumbigal. In Malayalam, we call this Kaduva Thumbigal. Okay. Because in Tamil, what do you call Kaduva? I think it's Puli, It's Puli, yeah. So these are Puli Tatambuchis. They belong to the family Gomphidae. This is also an exceptional family because I told you when discussing the differences between dragonflies and damselflies, in most dragonflies, the eyes appear close together. But in the case of this family, see the eyes are like those of damselflies because the eyes are situated on two corners of the face. Okay, and also they appear like tigers because they have yellow and black markings throughout their bodies. Okay, so this is the most common species of this family, the common clubtail, also known as scientifically known as Ictenogompus rapax. It can be identified easily by looking at its anal appendages. They're very large, very long, like a spear, and they have a flag-like appendage below it, the anal appendages. This is not shown by any other species found in South India. So with this, you can easily identify the Ictenogompus rapax. But in the case of females, the anal appendages are reduced, and you will not find this pronounced. This flag is there, this flag-shaped appendage is there, but it is not very pronounced in a female. So even in the case of dragonflies, looking at a female, it is not easy to identify to species level. Okay, so the next species is Paragompus lineatus, which is known as lined hooktail. Okay, in the case of this species, the anal appendages look like hook, hook that we use for fishing hook. So that is why it is known as lined hooktail. Okay, this is the male and the female. In the case of female, it is not well developed. And now we come to this family known as Macromidae. Okay, these are very large. These are large dragonflies, and the eyes appear close together, and they have a speciality there. If you look at their anal appendages, they appear like a trident. They have two superior anal appendages and one inferior anal appendage, like all dragonflies. But they are very sharp and pointed, and they appear like a trident. Okay, so this is one of the commonest species, Epophthalmia vittata, that you can see flying around most of the large water bodies. And the Macromia species, we have some 10 to 12 species in the Western Ghats, but not many of them are seen near wetlands. So we'll not discuss them. And then we have this family called Cordelidae. This is a very special family because it looks somewhat similar to that damselfly family that we mentioned, Caloptergidae, because they had this green-colored metallic plates. Like that's how they appear. They appear to be studded with green metallic plates on their body. But that's not the only specialty. This species is actually found in high altitude forests in the Western Ghats. But for breeding, we believe this species is dependent on wetlands, or at least they come down to low elevations to breed. We have records of that happening. So this is a species, despite being a forest dweller, it is dependent on wetland for breeding. We do have such species. And then we come to this very common species, these are called Nirmuttanmar in Malayalam. Libellulidae. Okay, so these are very adaptable and very species-rich family. No generalizations can be made because they show a lot of diversity. This is the most familiar species. I know many people. I mean, Balachandran sir would have described and discussed this because this is the migratory dragonfly. Okay, so this photo was taken from the cold wetlands here. So they are dependent on wetlands for feeding and breeding, and they follow the rains. That's how they migrate. Okay, it can be easily distinguished because you do not see any other dragonflies in such huge numbers anywhere. Okay, so that is the easiest way to identify the species. Okay, so then we have the trumpet tail. As the name says, its tail or abdomen looks like a trumpet. That is why it is known as a trumpet tail. This is the male, and the female is dull in color, but it retains the shape of the male's abdomen. And then you have something called Brachydiplax chalybia, rufous-backed marshhawk. There is a rufous colored mark on its back. That is why it's known as rufous-backed marshhawk. And this is how it looks. It's not very beautiful dragonfly, but it's like some ash has been spread on its body. That's how it looks like. But it is very common dragonfly seen in our wetlands. And this is the very special dragonfly that I want to talk about. This is Brachythemis contaminata. So this is a dragonfly that has to, that has had to, hear a lot of bad words, because it's not. Look at its scientific name, Brachythemis contaminata. Contaminata, because it is mostly seen in contaminated waters in huge numbers. They can be seen in huge numbers in polluted waters. But it is not. We should not blame the dragonfly. It's not its fault. We are the ones who cause pollution, and most of the dragonflies fail to breed in such waters. But this dragonfly has adapted to live in such water bodies. That is why it can be seen in huge numbers in such polluted water bodies, and that is what has earned its name, Brachythemis contaminata. It is known as a ditch jewel in English. Okay, so it can be easily distinguished. So the male is bright orange in color, and the red orangish color spreads to its wings also. But the female is duller in color. And then we have the red dragonflies. The red dragonflies that are very common in the wetlands. At one glance, they all look the same. But just by looking at their abdominal markings, you can tell apart the species. Look at this species, Crocothemis cervilia. In the case of Crocothemis cervilia, the abdomen is red in color, and it has a black straight line on the abdomen, right? It's a mid-dorsal line, black in color. But in the case of Rhodophthalmus rufa, this species, the abdomen does not have any black markings. Okay. And in the case of Urothymis signata, the black line is broken. Okay. So here the line is full, complete, and straight. Here it doesn't have any black line. Here the black line is broken. That is how you distinguish these three species. And in the case of this species, Macrodiplax cora, this is a very special species because most of the dragonfly species cannot breed in saline waters. Okay, even this cannot breed in seas, but it can tolerate salinity up to some level. That is why it can be seen in estuaries. Okay, but this species can be easily distinguished by looking at its legs. You see how the legs join, where the legs join the body, you can see this white patch that cannot be seen in any other species. And also in the case of Macrodiplax cora, the coastal glider, the black markings on the abdomen are dumbbell shaped. Okay, so this can be seen in the case of females also. Okay, so these are the females of these red dragonflies. Okay, don't be confused. Or even the females retain the markings as the males. Okay, so now we come to a species known as Diplocodex trivialis. This is a ground skimmer. This is one of the most common species. It does not really require water. It does require water to breed, but otherwise it can be seen away from water, even on tarred surface. In roads, on roads you can see them flying around, perching. They perch on ground. Not many dragonflies perch on ground, but these usually do. So it looks similar to the trumpet tail that we discussed, but by looking at its abdomen, the shape of the abdomen, it can be easily distinguished. And the ground skimmer does not have any thoracic markings, but the trumpet tail has very clear thoracic markings. Then we come to a species known as Neurothemis fulvia. It is called fulvous forest skimmer. It can be seen in certain wetlands where you have vegetation around them. When there is shade, this is a species that requires some shade. So it is usually seen in forested areas, but you do see them in well-wooded wetlands also. In the case of most dragonflies, the wings are plain. You see, they are transparent. But only a few species have their wings colored. This is one such species. So the male is red in color. It can be easily distinguished. The female looks like it has the color of rust. That's why it is called Thavidan Thurumban in Malayalam. Then we have something called Swamitumbi in Malayalam, that is Neurothemis tulia. It looks like a Sabarimala pilgrim ready to pack up and go to Sabarimala. That's why it's called Swamitumbi in Malayalam. Swamitumbi. Okay, so see, its wings are also colored, black in color, and it has like white patches at the end of the color. Then you have two red-tailed dragonflies. Okay, so these dragonflies that I showed earlier, they're completely red in color. Yeah, while in this case, only the tails or the abdomen are red or reddish in color, pinkish in color. Okay, in the case of brown-backed red marshhawk, the thorax is brown in color, but in the case of Orthitrum pruinosum, that is crimson-tailed marshhawk, the thorax is bluish gray in color. So that is one key to differentiate between them. And then if you look at their faces, this part is known as frons. We told it's nose. It's not actually nose because dragonflies breathe through spiracles on their thorax and abdomen. Okay, but this is actually face. It's called the frons, but it's similar to our nose. That's why I called it nose. In the case of brown-backed red marshhawk, the face or the frons is red in color. But in the case of crimson-tailed marshhawk, it is black in color. That is also a very easy key to identify. Okay, so now we come to a very common species known as Orthitrum sabina. Okay, this is like a large dragonfly. It is a very fierce predator, you see, and it usually prefers eating other dragonflies. See this? This is eating, this Neurothemis tulia, or the pied paddy skimmer. It's eating its head. Okay, so this can be easily distinguished because not many dragonflies in our part of the country are green in color. So this dragonfly is green in color with black markings on its body. Then we come to a dragonfly that resembles a butterfly because its wings are beautifully colored and they fly very slowly like a butterfly. They have a fluttery flight. Okay, so this is called Onathumbi in Malayalam because it is usually seen in August to September months. Now we come to the crimson marsh glider. It's one of the most beautiful species of dragonflies. You see, have you ever seen any organism with this color? What do you call this color? In English, they call it crimson. I'm not too sure. It's not just crimson that is there. You have red here. You have some violet here. So you do not find any other dragonfly with such a coloration. So it can be easily identified if it is a male. But the female, you see, it looks very different. It's yellowish and it's dull in color. Now we come to a species known as Kakaditumbi in Malayalam. It is known as long-legged marsh glider. See, in the case of all dragonflies, the hind legs are very long. You see, compared to the other legs, the hind legs are very long, but not as long as this, right? Okay, for in this species, that is Tritomis pallidinervis, the hind legs are very, very long. So it is known as long-legged marsh glider. And the male and female can be easily distinguished because it is the male which wears lipstick. You see, this bright violet colored lipstick-like coloration is seen only in the male. The female, in the case of female, the face is more or less plain. Now, we have seen some very common species that we may come across in wetlands. Now, we need to look at certain intriguing observations that we have made. Okay, so this is a species, Platylustes platystylis, that was actually described in somewhere in 1800s by Rambur. But it was believed to exist only in parts of Southeast Asia and in Bengal. But since 2017, there were observations in Kerala. There were photographs that were being shared in Kerala by this ordinate enthusiast called Raisen. So I teamed up with him and I went to his place and saw this species. We made this publication, but it was called this. Do you know the significance of the CF? This Platylustes is the genus and the Platystylis is the species. Okay, but this CF came in between because we are not sure about the species because for taxonomic purposes, we all rely on this book by Fraser, that is the Fauna of British India. In the Fauna of British India, Fraser describes that he's not sure if this belongs to all the specimens collected belong to the same species. So we are not sure if Platylustes platystylis is one species or more than one species. We were not sure during the time of this publication. But simultaneously, scientists from ZSI, Zoological Survey of India, were working on them. And they belong to two different species. One is Platylustes platystylis and the other has been named Platylustes kirani. Okay. So, and both these species are wetland dependent species. They are very rare and they cannot be found anywhere else. We have done surveys in forested areas. We have done surveys in other wetlands, but they are restricted to certain pockets of wetlands seen in Thrissur and Kannur districts. I mean, as far as we know. They may be found in other places, but we do not know what restricts these species to these certain microhabitats. So if you destroy these microhabitats, for by filling up wetlands or by pollution, we lose these species. And then there was a study, that we did. We compared the diversity of cold wetlands, diversity of alternates in cold wetlands with the ponds situated nearby. Okay, so we selected some 20 ponds and we selected some 20 sites in cold wetlands, and then we compared their diversity. And it was, it was very revealing. The results were very revealing because totally we were able to we were able to record 44 species from the wetlands, and we got only 30 species from ponds. And 14 of these species seen in wetlands are exclusive, which means they are not found in any other habitat. They are not found in ponds. But ponds did not have any exclusive species. That is, whatever species were seen in ponds were also seen in the wetlands. So this highlights the importance of wetlands as habitats for ordinates. And this is the reason why the wetlands are supporting such a huge diversity of ordinates. They have these all kinds of different microhabitats, while the pond has only a single kind of a microhabitat. It doesn't have so many microhabitats. So since the wetlands have different kinds of microhabitats, each microhabitat will have its own species assemblage. That is why the wetlands are able to support all this diversity. Now, what are the threats to these wetlands? And in turn, they are also a threat to the ordinate species also. So this is another photograph clicked from cold wetlands only. You can see that the wetlands are getting filled up. We are growing, we are increasing in numbers, and we need more buildings, we need more housing complexes, we need more shopping malls, and we do not have places to grow now. Especially in the case of Kerala and Tamil Nadu, we are running out of space. So we are encroaching into wetlands. That is the single most significant threat for the ordinates and the wetlands. You can see the wetlands being filled up here. And the increasing use of pesticides, pesticides, it includes herbicides, insecticides, rodenticides, everything is being used indiscriminately and unnecessarily also sometimes. That is also affecting the ordinates. So all the other problems that affect the wetland ecosystem also directly affect wetlands. So what can we do for conserving ordinates? These are some of the steps that can be taken. We need to conserve their habitats. We need the pond and wetlands, right? We need the fresh water for our own well-being and survival. So we just need to do that. We just need to conserve water bodies for our own sake, and the ordinates will get conserved automatically. And we need to conserve the remaining forests also nearby, because as I told you, there are species which depend on both these ecosystems. For foraging, they depend on forests. For breeding, they depend on wetlands. There are a few species like that. So if we are to conserve such species, we need to conserve the forests also. And we have to reduce the agrochemicals. So this is easily said than done, because if you have interacted with farmers, they will inform you that it's impossible. It's almost impossible to cultivate without using any chemicals. So that is something that we researchers need to address. How to cultivate and without using agrochemicals, or by minimizing the use of agrochemicals, and by ensuring a fair price for the farmers. Then we can actually enhance the remaining ecosystems by digging more ponds, or maybe planting more plants near the water bodies, so that the ordinates can happily breed there. So thank you for listening to me so patiently. If you have any doubts, I have deliberately kept my presentation short so that at least a few of you would be interested to interact with me. So this is something that I have shared with you already. This is in an ebook that you can use to identify species found in Kerala. I'm pretty sure that it will work very well for a species seen in Tamil Nadu also, because we share most of the species. This is freely available in our website. I'll share the link here again. And these are some of the other material that can be found in the website. Okay. Thank you. And I have shared pictures of not just mine. They belong to many people, and I thank them all. Thank you for listening to me. Now I am open to questions. Please do ask questions. Please, I'm waiting for them. Okay. Thank you. Am I audible? Hello?
Yes, sir, you're audible.
Okay. I'm open to questions now. I'll be happy to take questions. And I will also share the link of the ebook. Anything. You can ask me anything. Anything related to wetlands, anything related to ordinates, any project that you want to do. Okay, thank you for thanking me, but I would be happier if you ask questions, anything.
I think everyone is much clear about the session. Thank you, sir. Thank you so much for letting us such an informative and such a valuable facts for us. Thank you so much. With much pleasure, let me invite Ms. Gobika Devi M., our guest lecturer of Department of Zoology, St. Joseph College for Women, Alappuzha, for the vote of thanks.
Thank you, Ermina. Good afternoon to all. It gives me an immense pleasure to deliver the vote of thanks for this memorable occasion, the third day of our National Symposium, the Conservation of Odonates. First, I extend my sincere gratitude to the principals of both college, Dr. Ledadi Koto, principal, St. Joseph's College for Women, Alappuzha, and Dr. S.K. Jayanti, principal, Vellal College for Women, Erode. And I also express my heartfelt thanks to the SOS, the Society for Ordinate Studies, and Kerala State Biodiversity Board for their unflinching supports and coordination. I would like to express a hearty vote of thanks to our resource person, Mr. A. Vivek Chandran, conservation biologist, SOS, made an excellent talk on the diversity of odonates with focus on wetlands and emphasizes the different type of dragonflies and damselfly, odonates taxonomy, morphology and sexual dimorphism, threat and conservation of both wetlands and odonates. The explanation with presentation was very beneficial, sir. We are really enlightened your presence and your knowledge. On behalf of the both college, I convey my sincere gratitude, sir. Thank you. I also convey special thanks to the organizing committee and coordinators for making the symposium a fruitful session. Last but not least, I thank all the participants for their active cooperation for making the session is a resounding success. Once again, thank you all for their cordial cooperation. Thank you.
Thank you, ma'am. And thank you everyone for coordinating for such an informative session held up today. We'll wind up for the time being and let's meet tomorrow for another session brought up by Mr. Sujit V. Gopalan, expert consultant and conservation biologist, Society for Ordinate Studies, based on the topic Ordinates for Monitoring Ecosystem Health. And that's all for today. Once again, thank you so much.
Thank you.